My name is Imani Jenerette, and this is
Olexia Mitchelle.
the disease that we decided to do our article on for our assignment was myelodysplastic syndrome, also known as MDS. So what is myelodysplastic syndrome? MDS is a heterogeneous group of hematologic neoplasms classically described as a clonal disorder of hematopoietic stem cells leading to dysplasia and ineffective hematopoiesis in the bone marrow. The neoplasms are located in the blood and the blood-forming tissue like the bone marrow and the lymphatic system. The clonal disorder that occurs replicates multiple types of cells within the genetic mutation, which can develop into different types of blood cells. This disease can also be transformed into acute myeloid leukemia, also known as AML, and it's usually diagnosed in older patients. Lastly, more than 100 genes have been found to be recurrently mutated in MDS. In this picture, you're going to see Florence in situ hybridization, which is also known as FISH, which is a type of test that's done to see how chromosomes are looking. And in this one, there's going to be a probe for RB1 gene on a metaphase spread showed one RB1 signal on a normal chromosome 13 and the second clone. The RB1 protein acts as a cell cycle regulator, which blocks the transition of normal cells from the G1 phase into S phase of the cycle. The FISH test maps the genetic material in human cells more specifically so it can test specific genes or portions of a gene. In this figure, you're going to see multicolor blending, also known as MCB, which was applied to determine which chromosomes were involved in the chromosomal aberrations. Chromosomal aberrations are the changes in the structure or number of chromosomes. In this figure, with the chromosomes that are displayed, the chromosomes on the left are the normal ones, and the chromosomes on the right are the derivative of the left. The MCB stains the chromosomes, and the colors that appear describe the locations of the genes on a chromosome.
So G banding or Glimisa banding is a technique to use cytogenetics to produce a visible karyotype by staining condensed chromosomes. The characteristic banding pattern is able to be seen once the chromosomes are dyed and then looked at under a microscope, as we see in this picture above. The banding pattern can distinguish chromosomal abnormalities or structural rearrangements such as translocations. People with MDS might not experience signs right away, but they will develop over time. So things that can occur when a patient has myelodysplastic syndrome is unusual paleness, which occurs due to a low blood cell count, easy or unusual bruising or bleeding, which occurs due to a low blood palpitate count, pinpoint-sized red spots just underneath the skin that are caused by bleeding, frequent infections, which occur due to a low white blood cell count.
I'm trying to get a bunch of work done tomorrow morning, but I mean, I can.
So different subtypes of MDS syndrome. Myelodyplastic syndromes with single lineage dysplasia, that means the one blood cell type, white blood cells, red blood cells, or palpitates. NDS syndrome with multilineage dysplasia. In this subtype, 2 or 3 blood cell types are abnormal. MDS with ring cytoplasms. This subtype involves a lower number of 1 or more blood cell types. MDS syndrome with isolated del5q syndrome. People with this subtypes have low numbers of red blood cell types, and the cells have a specific mutation in their DNA. MDS with excessive blasts. In this subtype, any of the 3 types of blood cells can be affected. And then MDS with the unclassifiable In this subtype, there are reduced numbers of one or more types of mature blood cells, and the cells might look abnormal under a microscope. And that is it for our slides. And I think a really interesting thing that we found was like this plays a role in a lot of different diseases that we see throughout human history, kind of, because we have like, okay, how we see the different chromosomes, and like say with different number of things. So, but yeah, that's pretty much our presentation for today. I hope you have a good day and good night.